Hello, welcome. Can you put in the chat whether or not you can hear us okay? Oh, awesome. We got a yes. All right, we're just going to be waiting on some people to join, but as we're waiting, we've got a little poll to figure out where everyone's calling in from. I'm calling in from Northern California, up in the Redwoods in Arcata. Hey, everyone, welcome, welcome. Oh, we got a Mount Shasta, San Luis Obispo. We're going to give it a couple more minutes for a few more folks to join in. Thanks everyone for coming. All right, as we're waiting for some stragglers, we also have an important question because we're looking at soil biodiversity. We want to know whether or not you think a columbella or a tardigrade are cuter. So we've got that poll going. I think in this photo, a columbella is cuter, but I think in general, tardigrades. Yes, Kristen, I agree. Oh, we got 33 for tardigrade so far, 67 for columbella. Jan, vote for columbella. Awesome. All right, we're gonna get started as a few more folks might arrive. But first, thank you all for coming and attending. Um, my name is Colleen Kamoroff. I am a biologist with Stillwater Sciences, and we are helping support the California Institute for Biodiversity on this California All Taxa Biodiversity Inventory. And today we have a webinar specifically tailored for you guys, where we're going to go over our biodiversity sampling protocols. So first, before I get started on our protocols, I want to sort of introduce who California ATBI is and our mission, which is fairly simple, although fairly difficult to complete, and that is a complete inventory of California's biodiversity. So our focus is on the unexplored and undersampled biodiversity, and this includes the insects, fungi, soil biodiversity, and intertidal biodiversity. So across California, a lot of our vertebrate species and plant species have largely been discovered. However, the insects, fungi, soil, intertidal biodiversity, we're just at the tip of the iceberg with that. We're just getting started. So this is super important because California is a biodiversity hotspot. We are one of the most diverse species. We're one of the most places across the entire world. Although we're super diverse, we have huge monitoring gaps that exist for this type of taxon. This is for the insects, for the fungi, and the soil and intertidal biodiversity. And this is particularly important because these species can be highly endemic, sensitive to changing environments, and have a higher total species richness and abundance counts compared to the other terrestrial organisms. And these species also provide really critical ecosystem services. They sustain our food, they clean our air and water, they store our carbon, and they provide resilience against environmental threats like fire and erosion. So it's really important that we discover them and catalog them before we lose them. So how is CalATBI helping? So we are supporting fieldwork to find unexplored biodiversity, and we're doing this by putting boots on the ground, providing grants like the ones that you guys just received, also providing logistics and permitting support for fieldwork, and working with citizen scientist organizations. We're using this data to establish a voucher-based DNA barcode reference library, and this barcode reference library will be publicly available. We're also using this data as a baseline for future biodiversity sample comparisons. And the data can be used to help generate biodiversity distribution maps. Where in California are the areas with the highest biodiversity, and use that information to help inform conservation priorities and metrics. And lastly, we're preserving these specimens in collections. We're preserving a voucher of each one of these specimens for future generations and future research and technology. So how can you help? Well, you already are. We want people to collect biodiversity samples across all of California. And another way to help is using our sampling protocol and processing pipeline that we set up specifically for the California ATBI partners. So our sampling protocol's goals are to document as much biodiversity at each site that we're sampling as possible. So it's really important when you're going out there and collecting samples to think of the site with having a lot of different micro environmental areas. So you want to make sure you're sampling the microclimates, under logs, under rocks, and under different vegetation. And this way you can help document as many species as possible, and we can capture as much biodiversity per sample as possible. So I'm going to go over 3 protocols that we have specifically for the California ATBI to help collect this type of biodiversity. And these protocols include leaf litter, soil, and insects. So before I get started on the individual protocols, I just want to go over what I mean when I say site, because I'm going to be referring to sample collection at each site. So a site is an area of homogeneous ecotone. This is where the vegetation is similar. They likely experience similar environmental conditions. It has similar, like I said, similar vegetation. And each site is roughly, we're calling about a quarter mile. So we don't want sites to be collected, across a really, really large area, but you can collect it within a quarter mile of a central point and call it a single site. So in this image here, we have examples of 3 different sites. So site 1 in the yellow, that's just a meadow. So all the areas within the meadow would be considered one site. Site 2 is a forested area. And again, all the areas within site 2 would be a second site. And you are able to collect within that whole site and combine that sample into one and call it one sample for each site. And again, site 3 is sort of a riparian area. You're looking at sort of by the river and the rocks, and that would be a separate site as well. As you are looking to collect samples, we sort of want to avoid more disturbed areas, and this is specifically for the leaf litter and the soil collection. The insects collecting is slightly, it doesn't matter as much because we're targeting terrestrial insects that fly around all over the place. But for the leaf litter and the soil collections, we definitely get more biodiversity the further you get away from human disturbance. Each protocol has their own separate equipment. This is equipment that we have and we can ship to you in sampling kits. And these kits include a return shipping label. So everything's included in the kit. You just collect the samples, put it back in the box, and ship it back to us. So we try to make it as easy and simple for our partners to collect samples for us as possible. So getting into the sampling protocol, I'm going to first start with leaf litter. You can only collect leaf litter in areas where there is leaf litter, which is actually pretty few in terms of the biodiversity samplings that we've encountered so far. And specifically, we want moist leaf litter. And this is because the way that we process our leaf litter is through a Berlisi funnel, and that's where the insects actually crawl out of the leaf litter and into a jar of ethanol. So all the insects need to be alive for us to be able to process using this sampling technique. We have a simple way of collecting the leaf litter and then an even simpler way of collecting the leaf litter. The simple way of collecting the leaf litter is you rake up leaves directly into a sifter. You sift the leaves into a breathable cloth bag, and this is essentially just a pillowcase. And then you do this multiple times until you collect 1 to 3 cups of compacted leaf litter. The simpler way of collecting it is just putting some leaves directly into a Ziploc bag, as you see in this picture here. We do prefer the option of putting leaves into a breathable cloth bag. Like I said before, we're targeting living organisms in this method. So the breathable cloth bag just allows the insects to survive slightly better. However, putting leaves directly in a Ziploc bag and propping open the Ziploc bag so it does get some airflow, we do get living organisms that way as well. Because we're targeting living organisms and leaf litter is not a particularly forgiving substrate, we do ask that you ship these samples as soon as possible. So within 48 hours, ideally, although we have collected samples and shipped them within a week, and that works as well. Some other considerations is to make sure that these samples don't get squished or compressed, and then storing them until you can ship them in a cooler or a fridge is ideal. Again, we don't want to freeze these samples because we're targeting the living organisms. And like I should have mentioned this before, but we'll have plenty of time to go over any type of questions you have on each of these protocols once I finish the presentation. So feel free to hold your questions, or if you want to put them in the chat as we're going, we can address them as we go. Okay, moving on to the soil sampling protocol. Again, we have a simple way and then an even simpler way of collecting soil samples. Both of them involve digging a 15 centimeter deep hole from the topsoil. So that's it's not too far. We're just looking at the very little bit of topsoil. Collecting a 1 to 300 milliliters of soil samples from the hole and placing it in a Ziploc bag. And then repeating the steps 10 different times at 10 different locations. So this is where you really want to look at the site comprehensively and choose those locations that look slightly diverse from each other at your individual site. So you'd collect one location, by a tree, or another location under a rock, and that's why we can maximize the biodiversity that we're collecting at each site. You're combining all 10 samples into a single Ziploc bag, and that's just your one sample collected for that individual site. And this should be a 1 liter to a 3 liter soil sample in that Ziploc bag. The other option is just to collect a 500 milliliters of soil from 5 different locations. So this is a much smaller soil sample. We can use both these sizes of soil. If we get the 1 to 3 liter soil sample, we can do more with that. However, if we get 500 milliliters, we can also use that as part of our processing pipeline. These samples are slightly more stable than the leaf litter because soil is a easier substrate to survive in. But however, we do want these samples kept in a cooler or fridge if possible. if that's not possible because maybe your field work's working in the backcountry, or you don't have access to a cooler or a fridge for whatever reason, then they are stable, but try to keep them as insulated and as close to the ground soil temperature as possible. Again, we want these samples shipped as soon as possible because the quicker we get them, the more biodiversity we can pull from them. However, within 2 weeks is fine. We can totally get living organisms. And one of our processing mechanisms uses, it can pull out dead organisms as well. So the last protocol I'm going over is our insects collection. So we use a sweep net insect protocol. This is not necessarily the best way to collect insects, and we are totally happy to collect or to receive insects that have been collected in any sorts of ways. This was just the easiest protocol to use across a variety of our partners, and the easiest protocol that we can provide equipment for. So again, like if you're collecting insects in other ways, that's totally appropriate and that's totally great for this type of project. So our sweep netting protocol is fairly simple. You use a sweep net and sweep through. It's just sort of a long, butterfly net essentially. And you sweep through as much of vegetation as possible. You kind of want to go through the entire site, like I said, and sort of sweep comprehensively, against vegetation, grass, anywhere that you see insects. You aspirate the insects, which is essentially just breathing up the insects into a tube. Don't worry, none of the insects, you don't actually aspirate any of them, but this is an aspirator that you use. So you breathe up the insects using your mouth as a suction method to suck up the insects into a tube, and then you ended up spitting them out into a vial of ethanol. And then repeat the steps until you sweep netted the site for at least 20 minutes. Ideally longer because we do, again, want a comprehensive biodiversity across the site. We definitely want to aim to collect as many different species as possible. So if you see a certain type of insect in one area and different insects in another area, you want to make sure you sweep both those areas. And we really want to collect a minimum of 50 insects. And this is just because the processing of this is slightly laborious. So picking only one insect or one large insect into a vial and shipping it back to us is less helpful than having a bunch of small guys in the sample. These are stored in vials of ethanol. Excuse me. So they're fairly stable. They would like to be stored in a cool, dry location. The fridge is fine. Again, we don't want to freeze them. And then just ship to us when you can. Like I said, they're fairly stable. So if you ship them to us within 2 weeks, you can also collect them and ship to us after the end of a field season. That's totally fine. We don't want them sitting in your fridge for months and months and months. However, a couple months is totally fine. Okay, so I'm going to go over data collection real quick for all of our sampling protocols. And the way we collect data is using a Survey123 app. I hope this will work real quick. Oh. Can everyone see Imani? Can you let me know if you can see the ATBI Survey123? Awesome. Okay, so this is a very simple Survey123 form. You can use a web-based form where you don't even need a Survey123 app. However, it does work a lot better with the Survey123 app. It's free to download, and then you'll be able to download this survey form. It's all publicly available. So essentially, as you're entering data, the date time enters automatically. You can change that if you are entering data from a sample that you collected earlier in the day or earlier in the week. We want some information about who's collecting the samples, so that's your first and last name, as well as nice to have your email address in there, just so we can link the sample back to their collector. The next most important thing is to have the coordinates for where you're collecting the samples. This will automatically populate if you're using a phone or tablet. If for some reason the coordinates is not working, we do have some location notes, and that's where you can put in the UTMs for your location here. We have some information on collecting photos, and this is super important so we can qualify the environmental conditions of the site after the fact. And this includes a photo, sort of an overview of the site. We want a picture of the ground. We want a picture of the plants, the most dominant species, and any type of notable features. There's some optional information about the habitat. And then you will need to just collect, let them know that you're collecting a sample. Click what type of sample you're collecting. So let's say if you're collecting the soil sample, you collect soil, or if you're collecting the insect sample, you collect insect. And there's also one for leaf litter. There's an option to scan the barcode. So all of the equipment that we're sending to you will have a barcode on it. So you just scan the barcode and the sample ID will automatically populate. If you're collecting additional samples, you just press the plus button. And then if not, you can review the data, make sure it's all QC'd, and then press submit. All the Survey 123 form data ends up with us, and we're happy to share that back to you if that's information that you would like to have. And all the Survey 123 form, the sampling collection data will be linked to any of the processing and the results. So any type of occurrences or insects that we find in the sample will all be linked back to that collection data. So just some quick best practices while you are collecting samples. First, practice leave no trace. So this is particularly important when you are collecting soil samples. So if you're digging a small hole, it's really nice to disguise that hole, do a little bit of restoration work, make it look like no one's ever collected at that place. It's also nice to sort of limit trampling of vegetation if you're going out sweep netting. And then your own personal equipment and trash. Just pack it in, pack it out, just general best practices. For landowner permission, you need to make sure you have landowner permission to do the type of sample collection that you are doing. So this is generally not a problem if you're collecting on your own land or private land that you have permission already. However, if you're planning on collecting samples on federal land or state land, that does require additional permits. And some of those permits we actually have. So if this is something that you're interested in, we can potentially help with that permitting process, or maybe you'll be able to get onto our permits as well. Additionally, we have a scientific collecting permit for non-listed insects, and we were able to get people onto our scientific collecting permit. So if that's something the landowner needs to have for you to be on, let us know and we can include you on that permit. We also want to make sure that we're preventing the spread of diseases and pathogens. So make sure that you clean your soil sampling equipment between sites. I mean, the best way to do that is using a 10% bleach solution. If that's not available to you, just wiping off the dirt. If you are collecting some of the 3-liter soil samples, we have a detailed protocol that we'll be linking later on in this webinar, where would love for you guys to clean everything with 10% bleach solution, because we will be using a subset of that soil for environmental DNA analysis, and that does require having clean equipment. If you catch bumblebees in your sweep net, we ask that you clean your sweep net. Again, a bleach solution works great for this. We have additional information, especially if you're using our scientific collecting permit to collect insects off of. And lastly, we just don't want to be vectors for, again, disease or any type of invasive species. So make sure your clothes and your shoes and all of your sampling equipment is generally free of any type of debris or dirt or seeds. That can be particularly an issue in sweep netting. If you capture a bunch of seeds in your net, you just want to make sure you're not moving those seeds to a separate location. So how exactly can you guys sign up? Imani will put a bunch of references in the chat right now, but here, this is the Become an ATBI Partner is a form for you to fill out to let us know which protocols you are interested in sampling, and also how many sites you're planning to sample, and then whether or not you think you might need some permitting support. So all this information will be really useful for us. And once we get that, we can directly ship you the sampling equipment, or you can feel free to reach out to me directly if you have any type of additional questions, and myself or Imani, and we can help you through any questions you have regarding the protocol, our sampling, or permitting, etc. So Imani just put in the chat, it's a link to our detailed protocol. So I kind of just did an overview, but the protocols are detailed on both the soil sampling, the leaf litter, and the sweep netting. She also put in the chat a link to our Survey 123 form, which again is very easily to download onto your phone or tablet. And then also a link to this form, the CalATBI form, Become a Partner. So with that, I want to open it up to any type of questions that you might have. Feel free to raise your hand and I can unmute you, or if you want to put something in the chat, we can work that way too. It seems like no questions so far. All right. Well, if nobody has any questions, again.
Sorry, Colleen, we have a couple of people typing in the chat.
Oh, perfect. And while people are typing questions, I'll just put my email address in the chat. From how many sites would you like us all to sample? As many as you can. If you're going out in the field and collecting samples, we kind of want them all to collect them at all potential sites. And again, like your field site might incorporate multiple sampling sites. We've got a question from Andrea. If you are being in touch, she also fill out the form. Oh, yes, we've been in touch. Don't worry, I'll be reaching out to you directly. So thank you so much. You don't need to fill out the form. Rory's got a question directly related to your project. Okay, we'll get to you, Rory, but Kristen has a question of what permitting support looks like. That's a great question. So I mentioned one, we have an SCP where we can collect special status insect species. Part of that SCP has a citizen scientist clause on it where we can include people who are working as partners. We have a very easy quiz for you to take, just to make sure that you know what an insect is and some of the requirements of that SCP. So once you take that quiz, we can add you, send CDFW a list of the folks who should be added to the form or to the SCP, and then you can be added to it. You don't necessarily need a scientific collecting permit to collect insects specifically on private land. And I also don't think you need it for federal land. It's kind of more specifically for state land, but it's something that we have in case someone asks for having a scientific collecting permit. So that's one of the permitting supports that we can provide. The other one is land access. So you typically need, if you're wanting to collect samples on national forests or BLM land or any type of federal or state lands, you do need, excuse me, permissions from that landowner. And so we have a programmatic BLM permit that allows people working with us to collect these biodiversity samples across BLM. It does require some advance notification for some of the field offices. We also have some national forest permits. We're working on expanding those, but you have to go forest by forest, so it's slightly a slower process to get those permits. So if you're looking at national forests or BLM land to conduct this type of research, let us know, and we probably already have those permits in hand. Additionally, if you're looking at somewhere else and maybe want some advice on how to go about that permitting, like feel free to reach out. We can let you know where we're working on, what's available, and maybe any type of lessons learned that we have. And Rory, I can unmute you if you'd like to ask directly about your project. It might be helpful for other people who might have a similar question. Okay. I think you should be able to unmute yourself if you like.
All right. Thanks for the talk. I was going to do all the sample processing and sequencing and data upload myself. So is that still okay? Or is this talk seems more like just sample collection based? And I'm just wondering if I'm in the right place.
Yeah, no, totally. I think each individual project has different components. Some people are just collecting, and some people are doing the collection and processing as part of more of a comprehensive research project. And that's totally okay. I think where we want to see if there's any wiggle rooms, like you're collecting for your own research project, and that's contributing to the CalATBI. But is there any room for you to like help collect, excuse me, like additional soil samples so we can use them through our pipeline and to collect additional biodiversity across your sampling? Does that make sense?
Yeah. Is there some sort of, do you have protocols for that, or any sort of forms maybe for uploading sequences or something like that as well?
Oh, that's a really good question. We are developing that right now because we are developing what the voucher-based DNA barcode reference library is going to look like. So we don't currently have that information available, but we will definitely be reaching out once it is more established. And we do keep tabs of, you know, all the grantees who are collecting this data, because we do want to sort of incorporate your data into this larger database, whatever that ends up looking. And it might just be, you know, a download from Bold, like if people are already storing like these sequences in already publicly accessible databases. So again, that's something we're working on. We don't really have it set up right now, but we will be reaching out sometime in the future.
So you're still planning on setting up but so there's no database currently, is what you're saying, for CalATBI?
There isn't a database. No, that's part of one of the deliverables of the grant that we're working on.
Okay.
Yeah, so we're really focused right now on just sample collection and processing, and then an entire different firm is dealing with the database and the data wrangling.
Okay.
Yeah. Is anyone else? These are great questions. Okay, with that, I very much appreciate everyone for participating. If you have any questions, please feel free to reach out to me. I'd really love it if everyone could fill out the ATBI partner form. Let us know if there's any, you know, maybe it's the main part of your project where you're helping collect samples using our protocol, or maybe this protocol part can be just like a part of your project. Let us know. Like I said, our protocols are fairly easy, and they're also really easily adaptable to fit whatever type of capacity that you have in your research. So with that, thanks so much. I hope everyone have a great holidays, and reach out to us. Let us know how we can help and support your research, because we're very excited to have you guys as part of our team.